Steven, está Balance y está
Eduardo.
¿verdad?
Sí, están todos.
Ok.
Doctora, ya está la doctora Jaramillo del DEGI.
Ah, ok. Hola Lorna, ¿cómo estás?
Todo muy bien, saludos, ¿cómo estamos?
Bien, gracias.
Les felicito por el panel, está bien interesante. No sé si me puedo quedar las 2 horas porque estoy sacando algo que necesito para mañana, pero voy a tratar de estar el mayor tiempo posible.
Muchas gracias.
¿Cómo no?
Gracias, decana. Muy bien. Steven, can you pronounce again your last name so I will pronounce it correctly? Even though I know a little Dutch, but
it's a difficult one. Steven van Bellegem, but it sounds like from Belgium. So
okay, Van Bellegem.
Yeah, perfect.
Okay,
Thank you.
good.
Can you hear me fine, by the way?
Yes, we can. Thank you. Thank you. We're just waiting. It's 4 o'clock, so we wait a couple minutes and then for people to come in slowly, but hopefully we'll be start on time. I'll just wait a couple more minutes. Bueno, pues vamos a comenzar. Muy buenas tardes a todos y a todas. Bienvenidos a esta serie de paneles, de 3 paneles, titulada La Universidad de Puerto Rico investiga el COVID-19. Y nosotros, desde el Centro de Recursos de Investigación Interdisciplinaria y Aprendizaje Subgraduado, CRiiAS, en nuestro recinto de Río Piedras, que estamos adscritos al Decanato de Estudios Graduados de Investigación, queremos pues, compartir con la comunidad universitaria y el público en general los proyectos que recientemente fueran seleccionados para estudiar en detalle los diversos aspectos del impacto que tiene el COVID-19, no solo a nivel fisiológico, sino también a nivel social y a nivel de comportamiento. Tenemos pues el privilegio de contar con la gran mayoría de los investigadores de los 10 proyectos, en particular los investigadores principales de estos 10 proyectos que fueron seleccionados y subvencionados por una propuesta, vamos a decir, de fondos del gobierno de Puerto Rico para estudiar este tema. En el día de hoy, que vamos a estar enfocados en el tema de la evolución del virus, el impacto que aparentemente induce, y esto lo vamos a discutir más en detalle, de trombosis y potenciales tratamientos para el COVID-19. Y para eso tenemos a tres investigadores que son parte de estas 3 propuestas. Y el primero que va a estar hablando es el doctor Steven Van Belleghem, que es investigador postdoctoral del laboratorio del doctor Ricardo Papa, catedrático del Departamento de Biología de nuestro recinto de Río Piedras. Luego estará presentando el trabajo de investigación el doctor Valance Washington, que también es catedrático de nuestro Departamento de Biología. Y vamos a finalizar con la presentación del doctor Eduardo Caro, que es catedrático auxiliar del Departamento de Ciencias Farmacéuticas de la Escuela de Farmacia del Recinto de Ciencias Médicas. Quisiera recordarles más bien una solicitud que por favor todos los participantes desactiven su micrófono, más bien para que podamos tener una mejor experiencia durante el panel. Desactivar su cámara de video, excepto los panelistas, por supuesto. Evitar compartir su pantalla durante las presentaciones. Que por favor toda pregunta la hagan en el chat y al final de las 3 presentaciones entonces vamos a poder contestar estas preguntas. Y por supuesto mantener una conducta de respeto hacia los panelistas y los que hagan preguntas. Esperamos que luego del día de hoy, pues tenemos un próximo panel para anunciárselos que va a ser el próximo martes 13 de octubre. Y ese tendremos la presencia del doctor Julio Hernández Correa del recinto de Mayagüez, la doctora Loida Menéndez, la doctora Carmen Cadilla y la doctora Ana Espino, todas del recinto de Ciencias Médicas, pero que tienen proyectos separados que están asociados a los temas del COVID-19. Así que, sin más, quisiera entonces presentar al doctor Van Belleghem. Steven, if you want to put your presentation now while I present you. El doctor Van Belleghem obtuvo su doctorado en la Universidad de Ghent, en Bélgica, en el 2014, donde completó un proyecto sobre los aspectos conductuales y genómicos de la evolución de los escarabajos. Y el doctor Van Belleghem es un investigador postdoctoral en estos momentos, como les mencioné, en nuestro recinto de Río Piedras, trabajando con el doctor Ricardo Papa, donde se enfoca principalmente en estudiar la diversidad de patrones de color en las mariposas. Pero como ellos estudian la evolución de los genes, pues ahora está bastante interesado en el tema del COVID y gran parte de su trabajo implica comprender la arquitectura genómica de los rasgos fenotípicos y estudiar la estructura de la población y contemplar los mecanismos evolutivos. Así que, Steven, thank you for being here. And I really appreciate you presenting about what you are doing in the laboratory of Dr. Papa. Thank you.
Thank you. Can you see my presentation?
Sure. Sí, we can see it.
Okay. Well, good afternoon, everyone. Thank you all for logging into this COVID panel session, and thank you, Dr. Carmen Maldonado, for organizing this event and giving me the opportunity to present. So our group is one of the recipients of funding to study the pandemic, and what we want to study is the genetics of the SARS-CoV-2 virus, its DNA and its mutations, as well as how the strength of symptoms in people relates to genetic characteristics of those people. Or in more technical terms, we call this genomic epidemiology. But first, let me also introduce myself. You already did it very well. So I'm originally from Belgium, Belgica, with the capital of Brussels, and famous for chocolate and beer. And I obtained there my master's and PhD at the University of Ghent in evolutionary biology and genetics. And after that, I have worked as a postdoctoral researcher here at the University of Puerto Rico and also at the University of Cambridge in the UK for the last 6 years. My specialties are in evolution, population genetics, and using computer programming to analyze complex data sets, also called bioinformatics. And the last years, as you said, I've been mostly involved, or I've been mostly using my skills to study evolution of colors in butterflies. But now things have dramatically changed, hence some adjustments in the way that we work and what we do, and hence our attempt in applying our scientific experience to studying COVID-19. So again, the proposal we were funded for addresses two aspects. One aspect is to study the genome or DNA of the virus, which we want to do to broadly understand how the virus has been introduced on the island, how social distancing measures have affected genetic diversity in the virus, where new introductions come from, and whether the virus might be changing or evolving. And the other aspect of the proposal is to study the genome of its human host, to study whether genetic diversity in humans may hold clues to varying severity of the COVID-19 disease. Let me first also mention that this work is also a large collaboration with researchers from the medical campus at the UPR, including José Rodríguez Orengo, Abel Baerga, Filipa Godoy Viterino, and researchers from Yale Medical School who are really at the forefront of battling this pandemic. Nathan Grubau, Anderson Brito, and Joseph Fowler from the University of Yale. And this work is also only possible thanks to clinical labs that sample and store the viral and human tissues. Okay. So somebody still has their microphone on and it's creating a bit of background noise. Thank you. So first, about the genome of the virus. The genome is very small, consisting of about 30,000 base pairs or DNA letters, which is about 100,000 times smaller than our own human genome. And the virus and its genome is built to hitchhike on the available cellular machinery in human cells to reproduce itself. And one of the more important components of the viral structure is the spike protein on its outer surface, which is used to bind the cell receptor ACE2 and mediates the merging of the viral cell membrane with the human cell membrane, and by doing so entering our cells. And the ACE2 enzyme is present normally in cell membranes of lung and intestinal tissue, where it in normal circumstances hydrolyzes or changes the function of a protein called angiotensin 2 that is involved in inflammation and controlling blood pressure. But in this case, the SARS-CoV-2 virus hitchhikes or uses that protein to enter our cells. And since that spike protein mediates the first steps in the infection, it has been a strong focus for the development of therapeutic compounds. Now, this shows the functioning of the virus as we currently understand it, but a scary aspect is that viruses can evolve incredibly fast. And I would like to put this now in some perspective. So I did some literature research, and I found that from experiments, the mutation rate of RNA viruses can range from 10 to the minus 6 or 1 in a million to 1 in 100,000 per DNA position per replication cycle of a virus. So one DNA letter will on average mutate it mutate per million base pair every time a virus replicates itself. And as a replication cycle of a virus takes about 10 hours, and there can thus be roughly 1,000 cycles in a year, that means that the mutation rate per site in its genome per year is 1 in 1,000. So, okay, maybe that doesn't say that much, but consider that 1 milliliter of spit or sputum can include up to 10 million viral particles. That means that on average in that sample, every single site has mutated more than once. Now understand that a single infected person may carry up to 1 trillion viral particles, so 10 to the 12th. This means that a single person's infection can generate a lot of new mutations in the virus. To put this yet in another perspective, we humans have a mutation rate of 0.5 times 10 to the minus 9th, which means 1 in 2 billion mutations per site. So viruses, roughly speaking, mutate 2 million times faster. So in terms of mutation rate, 1 year for a virus equals 2 million years for us. And if we go 2 million years back in time, the human species didn't exist, and our ancestors were still hanging out with Australopithecus. But note, of course, that my extrapolations are fairly rough, and it does not take into account differences in population size, founder events, or natural selection. But overall, looking at the SARS-CoV-2 viral genomes that have been sequenced in the last months, the accumulation of mutations over time indeed closely matches the mutation rate found in experimental evolution of other viruses, roughly 1 in 1,000 mutations per site per year. So over the time span of a year, the average SARS-CoV-2 virus will have 30 mutations different compared to the virus at the beginning of the year. And here in the slide, it can also be seen that there are some hotspots in the genome of the virus where the virus seems to be accumulating more mutations. Now, regarding these hotspots, from the high mutation rates of the viruses, they're also extremely adaptable. This is, of course, because a lot of mutations are being generated all the time for natural selection to act on, but also because they have immense population sizes, as I mentioned before. And populations or organisms that have large population sizes will experience less stochasticity or chance events, and natural selection will be very efficient in them. And to me, that's a bit scary, because the bigger the pandemic gets, the more adaptable and hard to battle it may become. Think, for instance, of the flu virus, for which we have to get a different vaccine every year because it's so prevalent and able to adapt so fast. And now, within the last year, one example of mutation that has gotten a lot of attention, actually just in the last weeks, is this one mutation in the spike protein, which has risen to high frequency quickly throughout the entire pandemic worldwide, and is being discussed as making the virus more efficient at infecting people. It had a very low frequency in January and February, but now is almost completely fixed throughout the world. On a more optimistic side, people also discuss that seeing the importance of this particular site may make it a valuable target to develop therapeutic compounds. So all this background information, not just to scare you, but also to say that this virus might be with us for a long time, might be evolving, and that there is a lot of importance to sequencing and mentoring its genome. And what we plan to do in our study is to obtain sequences of the viral genome in Puerto Rico from nasal swabs for a large number of samples during the course of this pandemic, using sequencing techniques available in our labs. And the first thing that we would do with this data is build so-called phylogenetic trees that show the ancestral relationships between different viruses from different patients, as sort of a family tree, and using the mutations in the genome from the viruses. And such things have actually already been done over the world, and have given valuable information about how the virus is evolving and how it is spreading worldwide. And it showed, for example, thanks to efforts from the CDC, that there were at least 4 introductions in Puerto Rico, 2 relating to New York and Washington State, and 1 relates to Germany, and ironically 1 to Belgium. But note that I haven't gone to Belgium since August 2019, and also that there's a chance that these infections may have come initially from Belgium, but passed on their way through the US for a little while, but still show a signal of being related to strains in Europe. So we would like to do such a study more rigorously on the island for hundreds of samples throughout the pandemic to monitor where new introductions come from, for example, after a period when rates of the infection were low, such as in June and July, to study how long viral lineages circulate on the island, and whether new mutations have occurred and what they may do. And I show this final slide on the viral genome to show how good we were doing from May to July, and how quickly things changed when things opened up in July. This shows the number of positive tests relative to the total amount of tests done in different localities on the island. And this also shows where infection hives may have been in the past on the island, and we will try to understand, using our sequencing, whether they indeed drove the new infections after the quarantine restrictions were lifted. Now, quickly, we also want to sequence the DNA or genome of patients that have contracted the virus and have showed widely different responses. There's a figure missing here, I'm sorry. Yeah, I don't know why it's not showing up. But let me describe the figure. So remember that we know the virus binds to an enzyme called ACE2 in human cells using its spike protein, and this enzyme is highly expressed in the upper respiratory tract and in our intestines, and to a lesser extent in our lungs. And this supposedly relates to the progress of the symptoms starting in the nose and the throat, and later developing in the lungs, and also causing intestinal problems. And at a later stage, severe immune reactions can result in so-called cytokine bombardments that recruit a lot of immune cells, and that will cause general damage to our own tissues. But apart from the ACE2 enzyme, there's also other human proteins involved in the infection, such as the TMP-RSS2 and NRP1, which are involved in the fusion of the viral membrane with the human cell membrane. And difference in the amounts of at which these enzymes, such as ACE2 and TMP-RSS2 and NRP1, are present in a particular person may cause variability in the person's symptoms. And importantly, many genetic factors that could cause variability in symptoms are still unknown. And to better understand this, we want to contribute in sequencing the DNA of patients that have different symptoms to potentially identify new genetic susceptibility factors or monitor the frequency of known genetic factors on the island. And we will again do this using stored nasal swabs and blood samples with associated data such as age, pre-existing conditions, and blood type, and using a cost-efficient sequencing strategy for human genomes called DNA arrays or SNP chips. And finally, I want to note that what we are doing fits in a global initiative that will also increase the power of our study. The global initiative of NextStrain has already collected and presented thousands of viral genomes from all over the world, allowing us to relate what we find to the global patterns. And the COVID host genetic initiative has been collecting thousands of human genomes to identify additional genetic factors contributing to susceptibility and symptoms, and has found so far a number of genes related to immunity. These genes here are very undescriptive names, but the general conclusion or result would be that these are related to immunity. And the public availability of this data will also provide the platform and power for us to do this study in the island. And with this, I end my presentation. I thank you for your attention and look forward to any questions or suggestions you may have later.
Thank you, Stephen. That was very interesting and very scary too. But we'll have a chance to talk once we have all three of you discuss your projects. Well, our next speaker is my colleague, Dr. Valance Washington. Y ahora voy a hablar en español. El doctor Washington completó su doctorado en la Southern Methodist University en Dallas, Texas, y su formación postdoctoral en el National Cancer Institute en Maryland, donde estudió el sistema inmunológico innato y su papel en el cáncer. Fue durante sus estudios postdoctorales donde descubrió el receptor de plaquetas Triggering Receptor Expressed in Myeloid Cells, TREM, like Transcript 1, el TLT1, y completó los estudios iniciales que caracterizan el TL1. Ha pasado los últimos 14 años acá en Puerto Rico y 11 de ellos como colega de nosotros en el recinto de Río Piedras. Y actualmente su laboratorio estudia la función de plaquetas en enfermedades cardiovasculares, trastornos respiratorios y el cáncer. En la serie más reciente de estudios, su laboratorio ha delineado un papel de TLT1 en el sangrado de origen inmunológico. Y su laboratorio demostró que TLT1 media la transición suave de los neutrofilios del vaso al tejido, lo cual es importante para reducir el daño de tejido asociado con la lesión pulmonar aguda y el síndrome de dificultad respiratoria aguda. Así que, sin más preámbulo, tenemos al doctor Washington. Thank you, Valance, for being here, and we look forward to your talk.
So, can everybody see my screen? Can you hear me? No, can't even hear me.
We hear you and see the screen.
Okay, excellent.
If you want to go into the slideshow, you can do that, or just leave it like that. It's up to you, whatever works. Oh, there.
I was going to go with this. So, mi plan original es para dar mi presentación en español, pero el problema es cuatro llegó más rápido que yo. So, entonces voy a regresar para hablar con ustedes en inglés. Pero mi slide título está haciendo comprensión de la naturaleza trombótica de las infecciones por SARS-CoV. Eso es a virus, and so now I go back to English. Anyways, it's a virus, and Steve has gone into a lot of this already, so I won't spend a lot of time, but I do want to point out that the SARS-CoV virus uses the ACE2 receptor for cell entry, and why that's interesting and important for what I will tell you is that there are a fair amount of ACE2 receptors in the lung. So when a virus infects the cells, as you guys probably know, the cell now, it takes over the machinery of the cell, it makes the virus, and usually the cells will rupture, releasing a lot of virus. So this happens in the lung, meaning that a lung is a prominent site of infection. And what is key about this is that the majority of patients that die from SARS-CoV-2 infections, they die because of respiratory failure, something they call either acute lung injury or acute respiratory distress syndrome. And what happens in these diseases or these syndromes is, if you look at this figure here, you have on the left side is a healthy lung where you have alveolar cells, type 1 and type 2, this being in the middle here being the alveoli, and then the vessels surround these alveolus promoting air exchange. However, when you get viral infection, and now especially in these type 2 epithelial cells, which have the ACE receptor, this causes an alarm, which is usually recognized by monocytes. And the monocytes will cry for help, and the neutrophils will be the first responders. So the neutrophils come in to help fix the problem. However, in many cases, they cause more damage. And what happens is they come in numbers, they leave the vessels, they go into the lungs, and then they will destroy these walls. So your body responds to this, releasing lots of cytokines. And this now body response will now cause more damage. And what you get is leaking in the vessel. And this causes coagulation from platelets to stop this leakage. And what we find is that about 20% of the patients with clinical symptoms also have coagulopathies, meaning they have thrombosis in the lungs. And the sad thing about that is that the people who succumb to SARS-CoV are usually have association with this thrombosis. So when you think about this thrombosis, it's led by platelets. And most of us know platelets from the fact that they clot the blood. And if you look at this figure here, where you get a rupture in your blood vessel, whether it be in your skin or internally, these little platelets organize fibrinogen to form a clot and stop the bleeding. And when you look at a platelet, there are many things on a platelet. It has a series of tools to help it handle the situation. There are receptors that are on the surface of the platelet that help cause the clot. And then they have receptors that are on the surface of the platelet that cause the adherence to the vessel wall. Then they have granules inside that hold proteins such as cytokines and other receptors that also affect the lesions of the lesions and help the platelet stick. And what you get is this rolling and adhesion, where you have receptors such as von Willebrand's factor and GP1B that cause adhesion to the basement membranes of where the breakage is. And then you have 2B3A, which is an integrin, which binds fibrinogen and helps make a platelet clot. Now, when this works, you stop bleeding. But in the situations such as in the infections of SARS, and you get an interaction from this P-selectin with neutrophils. This platelet neutrophil interaction, when handled well, helps handle the situation. However, this often goes awry, and you get a release of cytokines and oxygen radicals that cause a lot of damage. And this often leads to something they call disseminated intravascular coagulation. And what happens is you get uncontrolled generation of platelet activation, and it can take one of two pathways. It can take a pathway where this uncontrolled generation of thrombin, which activates platelets, causes the consumption of the platelets, consumption of the fibrinogen, and then you get bleeding. But you can also get small coagulants that get stuck in the small vessels. And what happens, you get ischemic tissue, you get damage, and this could lead to multiple organ failure. And while here, you can get the bleeding, so it becomes consumptive, and there are key factors that become depleted, and bleeding may ensue. The other side of this is it might be hypercoagulative, and you will get clots, and you get organ failure. And in some people, you actually see both of these things happen. Well, platelets come from the bone marrow, and just like red blood cells and the white blood cells, platelets are made in the bone marrow, and they're released into the blood and the venous system, and then they'll go throughout your body. And this has been a long-standing paradigm for many years. But as early as the late 1800s, like 1864, 65, around there were people who were starting to measure differences in platelets in arterial and venous blood, and they were showing that the numbers were different, and they were speculating that platelets may also be made in the lungs. And it wasn't until 2017 where a lab in San Francisco were able to demonstrate that megakaryocytes, which make platelets, which are usually found in the bone marrow, can also be found in the lung. And what they've done here in this little figure here, they've shown that if you take a PF4 CRi promoter and put it in front of a GSP, which is a green fluorescent protein, the green fluorescent protein, and then you use this rosa tomato, you can show here that the rosa tomato is for the lung interstitium, but the green are megakaryocytes. And they have demonstrated that megakaryocytes are found in the lungs of mice. And what was interesting about these lung megakaryocytes is that they have higher levels of pattern recognition receptors, like the toll-like receptors, for example, than the bone marrow megakaryocytes. What is also very interesting is that these megakaryocytes, unlike their bone marrow counterparts, they have a protein or a receptor called class 2. And what is so important about class 2 is that it's found, class 2 is found only on a very small population of cells in your body. They are found only on the cells of the immune system. Dendritic cells, macrophages, B cells, and T cells. And it is through class 2 that CD4 T cells actually regulate how the immune system reacts. And here we have now found this it has been found on the megakaryocytes of the lung. However, these findings are in mice, and the questions are they in human, and if so, how relevant are they? So this was found in 2017. So that question remained until actually this year, when some people who were looking at COVID actually did autopsies. And if you look at these autopsies, you can see, this is lung tissue here. And what they did was a staining with the CD61 and von Willebrand's factor, which are found in platelets. And they are able to demonstrate that these large cells that are found here are megakaryocytes. So this was some of the first evidence that what we find in mice may also be happening in humans. But it opens up many more questions. Do the amounts of the megs go up in the infections, such as SARS, COVIDs, or influenza, or even sepsis? And if they do they behave the same? If you go from one lung disease to another that causes injury, do the amount of megakaryocytes rise the same? Do they have the same receptors? And are they like the lungs of the mouse, and can that be used as a model? But more importantly, how do we explore these differences in humans? Because I am sure if I ask any of you to let me cut you up and look at your megakaryocytes in your lungs, you would probably tell me no. So this arrives at the specific aims 1 and 2 from the grant that we had put together. So it became obvious from the literature that if you have megakaryocytes from the bone marrow are released into the veins, and you have megakaryocytes from the lungs, which is also released into the veins, but it's released into the pulmonary vein. So those platelets will come out on the arterial side. So we should be able to look at the venous side of blood and the arterial side of blood and see if we can detect differences that can be attributed to the megakaryocytes in the lungs and the megakaryocytes in the bone marrow by measuring such things as toll-like receptors and for class 2. And so this led to the first aim is to develop a single-cell sequencing platform for platelets and define changes in platelet phenotype due to the regression of SARS. So we can now get healthy people, and we can get people who have SARS or other forms of sepsis and acute lung injury, and we can compare this on the single-cell sequencing platform. Single-cell sequencing in platelets, as far as I've seen, has not been done yet, so we would be the first to be able to do this. We would then like to build on that platform where they were looking at the megakaryocytes. Instead of just showing, hey, the megakaryocytes are here, we should be able to stain those megakaryocytes for various receptors and transcription factors to get a window into how they develop and if they're different in the humans. And so this led to aim 2, is to analyze megakaryocyte and megakaryocyte markers in the lungs of patients that have succumbed to ARDS, and we would get a comparison with them, other diseases such as influenza, mycoplasma, and also compare that to healthy lungs in the fact that if we could get samples from people who have died recently from something traumatic that didn't affect the lungs, we could probably get a insight of comparison to the diseased lungs and the healthy lungs. So for the third aim, we wanted to look at the thrombotic nature. So platelets as a driver to lung inflammation. If you remember, when you were pointing out that the platelet-neutrophil interaction will help drive inflammation. And platelets also have things that they'll release. And specifically, our interest is this molecule we call trem-like transcript. So there was a study, and this what you're seeing is a representation of a study from cystic fibrosis mice. Now, cystic fibrosis is this disease where you have a mutation in a chloride channel that leads to excessive mucus in the lungs. And the mice represent this just as they see it in humans. And that being said, when they get infections, they seem to have problems overcome them and they become infected repeatedly. And what they've done in this series of experiments is that they have mice that don't have this molecule called, or this receptor called cystic fibrosis transmembrane receptor. And they demonstrate that if they induce a lung injury here, that if you're wild type having 2 copies of the gene, heterozygote, you get a certain amount of neutrophils in the lungs. But if you are missing this channel, you get an increase of neutrophils in the lungs. And also they measure protein leakage inside of the alveoli, and this is called the bronchoalveolar lavage fluid, or BAL. And they demonstrate that the mice that have this channel missing have a greater amount of leakage in their alveoli. So these cystic fibrosis mice have increased neutrophil infiltration and leakage into the alveoli during lung injury. So to take this a little further, to focus on what is actually causing this, we now have the ability to remove the cystic fibrosis transmembrane receptor from specific populations of cells. And what this group did, and this is from Ortiz Munoz, and this was, I think, 2019, they removed the cystic fibrosis receptor from either the macrophages, the monocytes, or the platelets. And what they demonstrated was that if you remove it from the macrophages and the monocytes, it's not much different than if you just had a healthy mouse with all the copies. This is called flox mice, so it's not removed. However, if you remove it from the platelets and they use a promoter called PF4, which expresses this CRiiAS and removes the leukocytes from specifically the platelets, you see the same inflammation that you saw in the mice that had it removed from their whole body. And what this says is that platelets control the intensity of the response. So we want to explore some of this, some of these things that have that the platelets release and some of the cytokines that are released. So in our laboratory, we are interested in this molecule called TREM-like transcript. And what we have demonstrated is that if you look at people who have acute lung injury from various sources, that you can use this TREM-like transcript, the soluble form that's released after platelet activation, as a marker for the prognosis of these of individuals. So we had 799 patients with acute lung injury, distress syndrome, and we demonstrated that the platelets, if you have low platelets, you're more likely to succumb to the disease, and that's been shown before. And we use this just to demonstrate that the population that we were looking at fits the common, what is seen worldwide. And what we demonstrate, if you look at soluble TLT1 levels at day 1 or day 3 after diagnosis with the ARDS, we can demonstrate that if you have higher levels than 1,200 picograms, which was the median, you're more likely to succumb and you spend more time on ventilation. And this is happening on day 1 and day 3. And to address this in the physiological side, we can demonstrate that if you give lung injury to mice, and you look at mice that don't have soluble TLT1, that if you give back TLT1, it can stop the bleeding, suggesting that this TLT1 has a role in controlling what is leaked into the alveoli. So from this, we've derived our AIM-3. So when you get diagnosed with DIC, there's an algorithm that you use, but this algorithm doesn't tell you whether you're going to bleed or you're going to have a hypercoagulative syndrome. And the SARS patients seem to have a hypercoagulative syndrome. So what we want to do is to determine the key thrombotic parameters of the SARS-2-CoV patients, compare them to healthy individuals, and compare that to individuals with things such as mycoplasma or influenza. We will take the plasma of these samples, look at the D-dimer levels, look at the levels of thrombin, look at the levels of soluble TLT1, and hope to be able to look at cytokines and see if we can revise this DIC algorithm to help physicians determine whether the nature of the DIC is more thrombotic or more hemorrhagic. So these are the 3 aims of the project, and I look forward to questions after the end of the session.
Thank you, Valance. Very interesting as well. Okay, so now y ahora vamos a presentar al doctor Eduardo Caro, que es catedrático auxiliar del Departamento de Ciencias Farmacéuticas de la Escuela de Farmacia del Recinto de Ciencias Médicas. El doctor Caro es egresado de nuestro recinto en química, en particular en química orgánica sintética, y que recibió su bachillerato en el 2008 y de ahí continuó estudios doctorales en la Universidad de California en San Diego, donde trabajó en el área de síntesis de productos naturales bioactivos y obtuvo un doctorado en química orgánica en el 2014. Luego de trabajar por un periodo como químico medicinal para la compañía Arena Farm, recibió una beca del National Institute of Health a nivel postdoctoral para hacer investigación en el campo de la química medicinal en el Scripps Institution of Oceanography en La Joya en el 2016. En Scripps trabajó en el Centro de Biotecnología y Biomedicina Marina bajo la mentoría del Dr. William Gerwick, estudiando aislamiento, síntesis y caracterización biológica de productos naturales derivados de cianobacterias marinas. Así que para los microbiólogos en la actividad. Y actualmente es, pues como mencioné, catedrático auxiliar de este Departamento de Ciencias Farmacéuticas. Eduardo, un placer que estés aquí y estamos aquí viendo cómo estudias este problema desde la perspectiva de tratamiento. Eduardo.
Ok, ¿me pueden ver y pueden ver mi pantalla?
Sí, te vemos y vemos tu pantalla, sí.
Perfecto. Yo también, al compás de los compañeros que hablaron antes que yo, voy a moverme al inglés, simplemente que hay mucha terminología y me gustaría también pues que se comparta y se transmita todo el mensaje de la investigación que estamos haciendo actualmente en la Escuela de Farmacia como parte del Departamento de Ciencias Farmacéuticas relacionado a cianobacterias marinas y otros productos naturales marinos. So voy a cambiar al inglés. It's great to be here. It's a real pleasure to be able to share a little bit of the research we've been doing. I am very much a very young assistant professor. This is only my 3rd year at University of Puerto Rico at the Medical Sciences campus. And so the title of the talk today is really surrounding marine algae as a source of inspiration to produce new antiviral agents for SARS-CoV-2. And so I think that was a wonderful introduction to not only the seminar series, but to this talk. But I'll briefly then go over my background in terms of more specific disciplines. And so my career as a chemist began at University of Puerto Rico under the supervision of Dr. Soderquist, where we were looking at organometallic reagents for organic synthetic transformations. And I quickly became really interested in organic chemistry as a field of study. I did a research experience during the summer in Casey Nicolau's lab doing total synthesis of natural products and analogs. And this is really where I fell in love with natural products. So combining both my background as a synthetic chemist as an undergrad and this new natural product experience, I pursued my PhD at UCSD under the supervision of Dr. Theodorakis, where I worked on 2 natural products geared towards cancer. And so this was mostly synthetic organic chemistry towards the total synthesis of this compound, but also we were trying to describe the chemical biology. And towards the end of my time there as a PhD student, really got involved in the biology of these compounds and actually did some cell-based assays, and we tried to determine some mechanism of action. And I really wanted to figure out more of this biological side and how we could apply it to developing natural products into actual therapies. And so as I graduated, I joined Arena Pharmaceuticals, worked as a medicinal chemist, learned a whole lot about how to really optimize organic compounds to be suited for human consumption and to really prove to be valuable therapeutic options in the clinic. And even though this was all preclinical, I really started to understand that there was a big gap. And this gap mostly was we had natural products chemists that were describing beautiful structures that had great specific bioactivity, but these compounds are not really made or designed naturally to go inside a human. And so there were some challenges here to kind of merge the gap between medicinal chemists that were doing structure-based libraries and natural product chemists who were really tied down to these really intricate structures. And so I went to this Scripps Institution of Oceanography, and I really was pleased to be part of a very interesting and unique program called the Center of Marine Biotechnology and Biomedicine. And there I was able to study marine natural products, specifically those from marine algae, and took under my belt a project on this compound called majesculamide D, but also was exposed to many other projects that varied from genetics to bioinformatics to isolation of natural products, and kind of dipped my toes into a little bit of these fields. And so when I moved back to Puerto Rico, I really wanted to take all of my background to build a research program that not only thought about synthesis and organic compounds, but also thought about natural products and medicinal chemistry and how we could use marine biotechnology to really inspire those projects. And so to begin this talk, a lot of us probably know this somehow, but for the general public out there, we really have pushed this thought about natural medicine and complementary medicine and how we need to be more botanical. But truly, more than 50% of our FDA-approved drugs are derived of or inspired from a natural product or a natural product source. And so if we think about the revolution now with medical cannabis in Puerto Rico, but California leading the way, we can think about two compounds that are very bioactive, and THC, which in the early 90s there was attempt to patent this under the name Marinol, but proved to be ineffective based probably on synergistic effects that really weren't understood at the time. And more recently, CBD has found its way into the clinic for the treatment of seizures, especially for infants and young adults. Probably one of the most classic natural product stories is the best-selling anti-cancer drug of all time, which was inspired by the biotechnological production from Taxus baccata, and where you can generate an intermediate of very high complexity, and through semisynthesis solve this challenge of access to Taxol as an anti-cancer drug. We also know that our microbe world is a great producer of natural products, and we can think of Streptomyces as one of those genuses that is incredibly productive in generating different kind of antibiotics, including some of the most potent antibiotics known in these beta-lactam kind of compounds. And in our other microbes, in terms of fungus, they're great producers of natural products as well. Trichoderma can produce cyclosporins, these kind of polypeptidic compounds that have immunosuppressive activity. And most recently, I would say in the last 30 years, we've really looked to our oceans for inspiration. We know that 70 to 75% of our world is covered in an ocean, and we think that more than 90% of our biodiversity is contained in our oceans, and there's much of this biology in terms of organisms to be described yet. And recently, saconitide was approved by the European FDA to treat acute pain as an analgesic agent. And this was derived from this really interesting sea snail that produces a cocktail venom of over 100 neuropeptides, yet one of them is the peptide that produces this analgesic effect and can be used as a therapeutic agent. And Puerto Rico actually is not foreign to these kind of stories. We have a great success story that even if we can't actually pin it on some Puerto Rican scientists, because this was mostly developed in the United States and then further by PharmaMar in Spain, we have a tunicate that originally inspired the isolation of these really nice anti-cancer compounds known as ET743. This is actually a picture I took at Hobos Bay in Salina, where we actually encountered this tunicate during some of our collection trips. And this was developed, as I said, into a really nice anti-cancer compound. It was the story of the first unaltered marine-based drug, now under the trade name of John Dellis. For those biologists out there, this compound works by an activation-elimination reaction that then conjugates to DNA material, and this DNA damage induces then apoptosis and cell death. And so this was recently approved for the treatment of soft tissue sarcoma, or for those that have relapsed ovarian cancer. And this was highlighted not too long ago by an article by Rosa Ortega in El Nuevo Dia, highlighting this story and the infinite possibilities that the ocean possesses to really bring compounds to light derived from these natural sources. And so our lab, based on my experience at the Center of Marine Biotechnology and Biomedicine, we still have an active collaboration with Bill Gerwick and his group there. We started with something we thought we felt a little bit more comfortable with in marine cyanobacteria. And for those out there, a lot of people who ask me, algae has been a long time misclassified. And so now we have an understanding that there's such a thing as macroalgae and microalgae, where microalgae is a cyanobacteria, given that's from bacterial source. And the only reason it was confused for a very long time, as you can see in the picture here, also taken by me at Salinas, Puerto Rico, and Hobos Bay, is that it looks like these tufts, these like nice filamentive algae tufts. But if you were to inspect this under a microscope, you can see that these disc shapes are really unicellular organisms that grow in colonies. And they grow in these filaments back to back, extending themselves to the point where phenotypically we can see these and observe these in the oceans quite easily. So these were two things that were really important to us beginning these kind of projects, was being able to identify the bioresource very easily by eye, by snorkel, but also having something that's complex enough where it interacts in the environment as a microbe. And in the last 20, 30 years, there was this review in 2015 that really summarizes the work. It's been a really interesting field to look into in terms of the producing chemicals that are being secreted by these cyanobacteria. And we believe that the difference in terms of those of macroalgae is that they may be optimized by billions of years of evolution, given that they're such an ancestral organism. And in recent years, it's shown that these structures are able to inhibit multiple biological targets with high affinity and selectivity. You can see lignobelin A, a cytotoxic agent, at sub-micromolar levels. We have sodium channel blockers in the case of Jamaicamide A. As you can see, the names of these compounds are really hold tribute to where they're described from. And so you can think of Jamaican or Curacin as Curacao or Jamaica. We have Curacin A, which we recently isolated and were able to de-replicate from a cyanobacteria we collected in Culebra, Puerto Rico, as well as in Cabo Rojo. And so how do these things look? And so our first effort to do this was, I was a chemist, so to me the chemistry wasn't that big of a challenge, or let's say as scary of a challenge. Definitely still a challenge, but to us the biggest challenge was, how do we even locate these cyanobacteria? And so with the help of Bill Gerwick and several other people around the island, we were trained on how to find these, and we were happy enough to find a super bloom. So given that these are communities that grow in a bacterial fashion, there can be nutrient concentrations or certain light exposures, or just variants in conditions that can produce super growth of these organisms. And I'm going to click on this quick video where Patricia Chardon, who's a colleague at UPR-Mayagüez with great freediving experience, helped us in the collection of this Symploca species of cyanobacteria, which we just collected by the bags. These are mesh bags that we're using to collect this organism. We can easily trap them and then bring these back to the lab. And so this was a kind of a unique experience to collect this amount of biomass, but you can see that these opportunities are available here in Puerto Rico. And so what do we do once we collect these? Well, we just take these back to the lab. We are able to extract these with liquid solid extraction using some kind of organic solvent, filter these, rotovap them down. The students are then taking on the project to fractionate these in different kind of methods. Here we have Will Dellis, a graduate student in the lab, doing a normal phase fractionation. Here is a short video of myself doing a reverse phase fractionation once we subfractionate, so we can generate on average about 40 to 50 fractions per extract. And this can get quite busy in terms of the rotovap used in the lab, but after rotovapping those, we can send those off for biological screening. But our part in terms of this whole project has really been using GMPS, a pretty unique and novel tool to figure out what is the chemical composition within these fractions and subfractions. And so the way this works, and I'm kind of describing this in a very general fashion, there is definitely a lot more bioinformatics and math involved, is that if you go to a supermarket and there's a barcode and you go up to the register and it scans, it's going to tell you it's Fruity Pebbles. And great, you get the cereal, it probably has a description of what the product looks like, maybe when it was received. All the information about this product is in this barcode. And so what we're trying to take advantage of, where this tool takes advantage of, is that if you do a mass spec, meaning that a molecule first flies and gives you its parent mass, and then that parent mass is then split up into fragments and stable fragments, given the right conditions to be able to replicate this, on average, it will give about the same pattern in terms of how it fragments based on what structure it has. And so in this case, we can think about the cyclodepsipeptide that we were able to isolate from cyanobacteria. I'll talk a little bit about that further in this talk. And so if we think about now having chromatography attached to this mass spec, we can separate, then have some MS-MS2 data. GMPS then can score these based on similarity. And using Cytoscape to interpret this data can now generate nodes. Now what we're looking at here, each node or circle represents an individual parent mass or an individual compound that's represented. And the line or edges that connect these now represent how similar they are. So technically this space is qualitative, it's not quantitative, but it allows us to quickly screen in terms of chemotypes these samples and generate chemical profiles pretty quickly. And so I'll go quickly over an examples because we really want to see how we apply this to COVID. If we run an LCMS, meaning a liquid chromatography that separates the compound based on retention rate, and then we do mass spec, we can profile cyanobacteria pretty quickly. And here we have 3 collections, one from Sony Beach, one from Punta Magueyes, one from Las Palmas and Manatí. And if we can see spectra 1 and 2, which are corresponding to Punta Magueyes and Las Palmas, are virtually identical. And we look at these masses carefully, we can see that these samples in terms of chemical production are pretty much the same. So we can assume pretty safely that these are the same organism. Yet the one on Sony Beach, even though very similar by microscope, presents a very different kind of profile. Now, if we dig deeper and we look into some of the subfractions, we can quickly replicate one of the samples in terms of Manatí, which is similar to the one in Culebra, and we can quickly assign structures of compounds that are in our mixture as these cyclodepsipeptides that have anti-cancer activity. And so we don't spend much time with these known compounds. We can really look at now these new compounds, and let me turn on my pointer very quickly. We can take time in terms of these really small nodes that are not present there that we think that are structural analogs that have not been described just yet. And we can also look and send at the Sony Beach sample and say, well, there's new clusters that are not identified that we can't replicate. In this case, the size of the node of the circle is describing the abundance of this ion at 1696 in terms of its retention rate, and we can then isolate new compounds that are not there. And so this all begs the question, why have I talked about all of this so far? Well, we really weren't into COVID research. And the reason we got here is that during the lockdown, I was reading way too much. And probably a lot of you guys were as well. And as I was reading, I came across a very interesting paper that allowed me to relate how these algae may be suited to intervene or interfere or somehow inhibit mechanisms specific to SARS-CoV-2. And so the first paper I saw was this virtual screening project that had been published based on marine natural products. And I won't get into much details because they do a very nice kind of filter here. But basically, they look about over 14,000 marine natural products. They fill for a specific pharmacophore, which they believed could interfere with the main protease of SARS-CoV-2. And I'll talk a little bit more about this main protease and what consequences it has within the virus. And after the virtual screening and simulation and redocking, they generated some promising structures. And this one sentence I was able to extract out of this marine drugs paper is that the most promising inhibitors based on the screening effort towards this main protease were represented by a class of molecules specific to brown alga of the class of sargassum. And sargassum is a problem we have in Puerto Rico. We've seen it washing up for the last 5 to 6 years in huge amounts. And so this was the first indication I got that maybe we were onto something. But even more importantly, a paper in Science came out describing not only the structure of the protein, the main protease of SARS-CoV-2, which is incredibly important in maturation and in many kind of cleavage sites of other kind of proteins that are specific to the metabolism of SARS-CoV-2, but it showed that there was a ketoamide kind of compound that was able to fit within a cleft that would inhibit effectively this main protease of SARS-CoV-2. And if you look at the structure, the first thing I thought about was a paper we had published on another natural product that I had worked on as a postdoc of this alpha beta unsaturated kind of Michael acceptor with a polypeptidic ketoamide kind of structure. And so this immediately clicked in my brain and said, these compounds are very similar to those produced by cyanobacteria. We have some virtual docking studies that kind of give us an idea that, or a hypothesis that also brown algae can interfere. So why not just look at marine algae in general and make a biased library? And so that's kind of what we proposed. The big picture was, instead of making it a very open, big library with many kind of compounds, we really wanted to be biased on our collections. And our library was already pretty biased, given that we were working with cyanobacteria and some other kind of macroalgae. And so our whole point was, let's go snorkeling, let's get more of these organisms, but very specifically brown algae and cyanobacteria. Let's generate some extracts, let's fractionate them, and let's throw them at COVID. Now, this is easier said than done. I'm going to move quickly through the next couple of slides. The first thing that we needed to figure out is, if we're going to do this on a high throughput scale, we need to think about sample collection. and how are we going to track our sample collection? And I have a very talented postgraduate student who finished her undergrad in marine biology, and Gracie just basically figured out a tracking system where every time we do a collection and add it to our database, it automatically populates this map. If you were to click on a map, it says where we collected it, the GPS coordinates, what are those collection codes, what was our collection method, and this is attached directly to our database. And this is of real importance, because if we are to find a hit and we need more of this organism, we should know exactly where to find it and under what conditions we found it. And so what we did is basically a hypothesis-driven expansion of this marine natural product library that was going to be biased. Gracie is truly a pioneer and really pioneered this effort with me. We went out and collecting during COVID lockdown with permission of local authorities. We did some kayaking because we had no access to our boat, collaborators that have boat access. And so we actually got a pretty good workout as well. You can see here, this is about a 6-kilometer kayak to collect at 3 different sites inside Hovos Bay. And so cyanobacteria grow in very different shapes and forms. We've collected from intertidal zones, shallow waters, deeper waters, you name it. And so we've taken these back to the lab. They look very different. We tag them, we give them collection codes, we put them in Ziploc bags. These go straight into the lyophilizer to preserve the chemistry at a very cold temperature while drying the biomass. That dry biomass then goes into a very expensive and elaborate instrument called the Ninja. We blend that up and throw them in vacuum bags, and those go into the freezer until they're ready for extraction. And so we had already talked about how we do that extraction. We haven't really changed much about how we do this. We extract, we fractionate, and we send these now for our biological screening. And to get to that, we have some wonderful collaborators at Wistar Institute. I am not a biologist. I really work at the interface of chemical biology, where I understand biological data if it's explained to me. And we kind of look at what the structure can do, or what we can do to the structure to really optimize this biological activity. And so we have 2 kind of high throughput assays. Dr. Ian Tijen and Troy Messick at Wistar Institute are kind enough to run these assays for us. They are sub-awardees inside our COVID-19 grant. And one of them is based off this ACE2 spike interaction, and where if this interaction is disrupted, we lose fluorescence. And we also have a high throughput enzymatic assay based on the main protease, which we talked about as very much a main player in maturation and most of the metabolic features of COVID. So we're really excited about this. We just sent about 50 samples their way of crude extracts. And so now we're working against the clock to fractionate those and also to do all the chemical analyses on these extracts. So once they get the biological data back, we can put those together. And so we also have a great platform based on my experience synthesizing these compounds to really go to hit to lead. This is really important if we think about these being any kind of drug candidates or being pushed into in vivo studies. And so we do have a great handle in terms of being able to modify the ketoamide kind of structure based on our majesculamide D experience. And so with that, I think I've kind of gone a little bit over our just tight of time. I want to acknowledge our collaborators at UPR Mayagüez. Patricia has helped a lot in our collections in different areas of Puerto Rico. Dr. William Gerwick continues to give me support after my postdoc and has guided a lot of our experience now collecting in Puerto Rico. Dr. Ben Naiman, Kevin Tidgewell, Skylar Carlson have also been in collaborative efforts in terms of describing different projects based on natural products of cyanobacteria in Puerto Rico. I also want to thank Angel Diepa, who gave us a clue to where cyanobacteria grow within the Hovos Bay Marine Estuary and gave us permission to access that by kayak during the lockdown. And also the funding sources, RCMI seed monies, who helped start my lab. Sea Grant of Puerto Rico also contributed to seed monies to start my lab. And obviously the UPR COVID-19 Emergency Research Project Program for funding this awesome work. And I especially want to thank these guys. These are real troopers. They've really stuck with our research program. Sorry, Angel, I had to kind of Photoshop you in there because you weren't able to collect with us on this day. But Gracie has been a real hero in terms of going collecting with me on these really extensive collection trips, which may seem fun, but are a lot of work. Wildelis Marie and Chris have really been at the forefront of extractions, fractionations, processing samples once we get back late from these collection trips. And Juliette and Angel are actually our synthetic chemists in the lab as undergraduates, and they've been doing a wonderful job, receiving their training and doing really well with the Majusculumide project. And with that, I would like to close, and any questions, I would be glad to answer those. And please do follow us. We're on Instagram, on Twitter, and we also have our website that has a blog where we update what are we doing. And we sometimes do some Instagram lives about our collection trips.
Excelente, Eduardo. Gracias por esa tremenda presentación. Me encanta ese entusiasmo y de todas las cosas que estás haciendo que tienen tanta relevancia para nuestro entorno geográfico. Imagínate, las playas, las bellas playas de Puerto Rico que pueden tener tal vez la solución para este problema a través de los productos naturales que tú encuentras. Muchas gracias. Bueno, ahora tenemos un tiempito para poder hacerle preguntas a los panelistas. Tengo ya una para ti, Eduardo, ya que para que no te vayas, que pregunta la doctora Meléndez. Ella dice, how does this massive plant sample collection affect our environment? Can these compounds be synthesized in the laboratory?
Yes. Well, first of all, their impact. So we've applied for permits through Department of Natural Resources. We had conversations with different people from the marine environment. So the first thing I will kind of point out is that usually cyanobacterial growth on reef environments especially is a sign of bad health. And so right now, I don't know if you've read some of the things that have come out in the newspaper that we've had cyanobacterial blooms, and these are extremely harmful. And so there are such a thing as a harmful cyanobacterial bloom. So as we are collecting this, harmful algae, we are also kind of suiting our needs in terms of the lab. But we definitely have a handle for synthesizing them, and yes, they can be synthesized. And the whole goal is to have a synthesizable target at the end, because this is the only way we're going to be able to manufacture enough compounds to take this into preclinical development.
Muy bien, gracias. ¿Alguna pregunta del público? Yo tengo una pregunta. I have a question for Steven. Steven, what about, have you are looking at epigenesis in these events? Can we see the role of epigenesis in the mutation of the genome of the virus?
Well, if you tell me epigenetics, I would think of the human host part.
So that shows that I don't know.
Well, that is wrong, but I do not know that viruses would have epigenetic effects on their structure of their genome or the expression of their genes. I would assume that epigenetics can have a role in the sense that people with a different health background or people that smoked can have different genes being expressed in their lung cells.
Okay.
People with obesity seem to react differently to the disease, and to me, those could result from epigenetic factors.
Okay. Yeah. That I thought of that, so that's why I was asking. Okay. Yes, and then I have a question for Valence about the hypercoagulated syndrome that you mentioned that COVID induces. So the answer will be anticoagulants. At what stage should we give anticoagulants then for COVID patients? Valence.
They're usually given right as you get into the hospital.
Okay.
In fact, if you talk to some physicians, they'll tell you to take aspirin if you are diagnosed positive. But
okay.
Usually they'll give you low molecular weight heparin, and there's a discussion, an active discussion, where they use low molecular weight heparin or unfractionated heparin because one of them you can reverse. The other one you can't.
Okay. There's a question that Stephen is for you as well. He says, I didn't completely understand the goal of using scRNA sequence for platelets. Do expect different cell types in platelets, and why?
Well, it goes back to the point of having megakaryocytes in the lung and megakaryocytes in the bone marrow. So the ones released into the bone marrow will come out into the vena cava, and the ones, so you should be able to take the venous blood and take the blood that comes out of the aorta side and see if we can identify differences, because the ones that come from the lungs will populate the arterial blood, and you should be able to identify different populations.
Okay. Any questions from the audience that would like to unmute yourself y pueden hablar, hacer preguntas en español y en inglés y en holandés y en francés? ¿Alguna pregunta? No sean tímidos. ¿No? ¿No hay más preguntas? Entonces, pues yo tengo otra pregunta para los tres. How do you see at the end of this year, when funding ends, how do you see making, in addition to, contribution to the field, but do you see any way of creating collaborations with other laboratories in each one of in addition to your collaborators? How you see your work going beyond these collaborations? Where do you see yourself at after a year?
Well, I'll start because I was the first presenter. But I think we have the close collaboration with NextStrain is really with people that are at the forefront of battling this disease. They are developing new tests. They are storing and making public the data that is being used to find therapeutic targets. And the data that we generate very well fits into their framework. And I think that will pass on to beyond this single year of work.
Great. Balance? What?
Yeah. Well, focusing on aim 3, first we'd like to see if we can identify a way to help physicians better interpret the coagulation cascade. So what they're doing now, they say a person has a certain score, or DIC score. We know that there's disseminated intravascular coagulation. However, they can't tell whether it's a bleeding nature or a coagulative nature. And so the treatment would vary. Do you give platelets? Do you give heparin? What's the best way to treat that? And as far as looking at the megakaryocytes, we're looking at better ways to manipulate them. And if they have class 2, which my understanding of the pipeline, there's a paper coming out, really big paper coming on how they can affect class 2, it becomes important to have ways to understand how to evaluate this in humans. So we're hoping to be able to give a preliminary roadmap to understand if there are differences like in humans as we see in mice, and I have ways that we can identify these differences. Great.
Eduardo, how do you see el próximo paso después de ese año? ¿Qué tú crees que
Yeah, I mean, it's really difficult to know, especially in the drug discovery and development world. We can have massive hits and have great luck. And this can be developed into a much more like an NIH R01 project, or it could be a massive failure. And so we kind of have to be prepared for both. I think at the end of the day, if we can make a contribution to understand a little bit more about how we can develop retroviral agents that we need, because regardless of the vaccine or retroviral agents, we will probably need both and a cocktail of both. And we will need broad spectrum retrovirals. And I think the main protease is a great way to start. It's not the first protease inhibitor on the market, so this kind of gives us a little bit of hope. But just if we can make a global contribution to knowledge of archetypes, just structures that could potentially inhibit this main protease, I think we would be happy. At the end of that, I would love to think that in Puerto Rico, we can identify more collaborators to continue work on this scale. But yeah, it's hard to say. It's really hard to say. So I think we're lucky to be in this situation, to be able to contribute and do such interesting research. And it's been a real pleasure to participate of this kind of seminar and see what other researchers are doing, because now I have like 20,000 more questions to look into.
That's right. I'm glad that that has happened. And I hope that the next two panels will be the same, as interesting and as enriching for understanding the biology and potential treatments for COVID. Así que terminamos. Creo que no hay más preguntas y estamos terminando más o menos como pensé, como a las 5:30. Así que para mí siempre es un placer. Las presentaciones fueron pues, excelentes y exhorto a los colegas a que regresen el próximo 13 de octubre cuando tendremos a otros panelistas hablando de sus proyectos que son igual de interesantes. Así que thank you so much for coming. I really appreciate you being here. Muchísimas gracias a los tres. Y nada, nos veremos el próximo martes 13 de octubre. Así que tengan excelente tarde. Buenas tardes a todos.
Buenas tardes, gracias.
Bye. Bye. Saludos a todos. Gracias por estar aquí. Gracias.